As you have just heard, we're going to record the talks today. Those will be available for our folks and for the Duke team to also listen to later. And our first talk will be someone who really needs no introduction, Dr. Mike Lynch from Duke University. And sorry about that.
No worries. So if I can get the goal today. So Matt and I talked about doing the symposium to kind of bring the two groups together, which was a great idea. And he said, well, you should give a talk. And then the question was about what, right? So rather than sort of go through a lot of the data, which I hope that hopefully most of my lab and most of you guys have read all the papers, at least the technical papers, related to DMC technology and at least some of the patents you would hope. And in light of the fact that we have an audience that's largely not, it may not be as technical across the board, I thought I would share some thoughts and slides that are really more around a discussion of what's next in the field of biomanufacturing. That's a term that I'm using on purpose because if you're aware, there's sort of a new initiative from the top down in the White House to advance biotechnology and biomanufacturing in the States, right? So whether that's synthetic biology or metabolic engineering, the term of choice that will kind of be the buzzword for funding in the next probably 5 years, assuming that the regimes don't change, will be biomanufacturing. So we're going to use that term. Which is nice because it's actually a pretty old term, right? So, okay, great. So, biomanufacturing 6.0. We'll talk about the 6.0 in a second because that is not a sexy term that we should be using long term. So one of the points of discussion here is if DMC in our lab is going to take a leadership role in sort of defining the next stage of what is biomanufacturing, which I think we are poised to do. How are we going to lead that conversation and what are we going to call it, right? And so the preface here for the grad students and postdocs. In our group, we focus a lot on the value of storytelling, probably more than they would like, I think, in general. So, there's a lot of rules we go over of how you craft the talk, et cetera. They may think it's a lot more like this than the other slide in terms of a lot of negative feedback and argumentation and not feeling very good about themselves when they leave the room. But in general, I think they end up doing this pretty well. So I think it's working in that sense. But there are tools and rules for good communication. And I'm going to break a lot of the things I tell you to do in this talk. So do what I say, not what I do. Okay. So back to this conversation. So biomanufacturing 6.0. So some of the agenda items to go over today are sort of how do we define the next generation of biomanufacturing, right? I think that there is a lot of voices out there that are competitive in this space. More syn bio, as Doug said, which is how do you define that? I mean, some people define it different ways than others, but it's a very vague term. Metabolic engineering is also probably has been diluted over the years in terms of what that actually means. And so there's also machine learning and automation and all those types of voices that say what the next level of next generation of biomanufacturing should be. And so I think the question is, do we agree with that? Do we not agree with that? What is our vision and how do we sort of propose that to be sort of leadership in the field? So a historical perspective on metabolic engineering, we'll try to go through that pretty quickly. The two-stage dynamic metabolic control technology in context of that history, as well as sort of in context for the next generation biomanufacturing. And then a discussion, hopefully more of a discussion than a talk on sort of what do we do strategically, not specifically with experiments or research plans, to really make this future a reality, right? So why biomanufacturing 6.0? So this is actually adapted from a paper in Journal of Industrial Microbiotechnology in 2017. So there's obviously, pre-modern biomanufacturing, bread, beer, cheese, wine. 1910, you think about some of the things that we do, like ABE processes, they had their roots way back, right? We did some fermentations for chemicals back in the early 1900s. 1940s is when we started to look at taking strains that made things of use, like penicillin and other antibiotics, and more primary metabolites, and actually scaling up fermentation. So there was a lot of fermentation science there. There was actually a lot of fermentation science developed in World War II in response to a lot of the conditions that arose in that time. Biomanufacturing 3.0, so that's when we had recombinant DNA, and that's where we had insulin and enzymes and a lot of protein expression. And then sort of in the late 80s, early 90s, which we'll get into a little bit later, is kind of when we started doing pathways, and we could make metabolites with heterologous expression. So you have biomanufacturing 4.0 in 2000, which is sort of mammalian cells, hex cells, CHO cells, tissues, advanced biofuels, advanced chemicals. And then 5.0, which I'm adding on to this paper because this paper was pre-2020. Sort of people think about what's in the space, so synbio, CRISPR, gene therapies, CAR-T. There's more bioactives, and then non-natural chemicals are sort of a focus of a lot of the different groups, right? So what's next, right? So if we're thinking about, you know, not what are we going to do next year, 2023, but sort of where is this field going, and how do we make the biggest impact we can, right? So some people, and our lab has sort of two different focuses. One is sort of more on healthcare. So a vision of unlocking next generation healthcare is sort of one focus of, you know, what's next generation biomanufacturing and bioengineering. And of course, the other one, probably more pertinent to DMC in this conversation, is sort of a vision of a global sustainable bioeconomy, right? So defining what that means, first of all, is always a fun thing to do. But then how do you realize that, right? So is that zero waste? Is that, you know, no greenhouse gas, a combination of the both? So how do we make our grandest visions of from biomanufacturing a reality? So this is actually for you guys to join in. So you can go to this web link or text and try to answer this question. So I want to poll the room and figure out, you guys are kind of in the space, hopefully, or at least have a pretty good handle on the space. You know, what do you think, as a group of people, is really needed for next generation biomanufacturing? And I do not see any. There we go. I did counts on purpose rather than percentages, so I know how many people are not participating. I think we have 25 folks in the audience, so I'll give it a few more minutes. Okay, so I'll lock in what people have said. So the first thing to discuss is, and this happens a lot when I talk about what do we need. It's very diverse, right? There is no agreed upon discussion of what we need to kind of move the field forward, right? So we have systems engineering, advances in synthetic biology, advances in machine learning. We have no votes for to be invented technologies, and we have high throughput technologies kind of winning out. So if someone said high throughput technologies and is willing to volunteer and answer as to why they said that, they can maybe be brave and comment why. Anybody?
I said that.
Sure, go for it.
And I said that because the space that we need to mine, if you will, is very, very large.
Okay.
The number of possible combinations, you know, is astronomical, literally. So high throughput can only help us mine more of that space. It's not the only thing.
Fair enough. Is anybody else that said that say it for a different reason? Yeah.
Well, I was thinking about it from a manufacturing perspective.
Okay.
Higher productivity, which is how I interpret it.
That will enable better performance and lower cost. Got it. How about protein and enzyme engineering? I know probably one person who said that from my group, maybe not. Why did you say that? Anybody willing to say why they said that was the most important thing? Okay, well, let those four people slide. How about gene editing and genome engineering? Oh, sorry, yes, in the back, yeah. Okay. So if I can summarize that, specialize meaning make novel things, make new things that don't exist. Okay, I can get behind that. What about gene editing and genome engineering? What were the thoughts there? Yeah.
Is there anything more powerful than being more downstream?
Fair enough, fair enough. Anybody, so there's another person who said that. Yeah.
I agree about that. When we know the genome sequence, at least I thought we know everything is there, but obviously not. So only through the media feels like only through the editing and engineering you start to understand what is the relationship of all of those sequences.
Got it.
And only then you understand.
Got it. Okay. How about the people who said advances in machine learning and its applications? And probably people who've talked to me know how I feel about that one. So, Mikey.
I just feel like all of these are pretty important, but AI is kind of a very upstream application that you can apply to all of these. Like people have done machine learning for enzyme engineering, people have done machine learning for better gene editing design, all that kind of stuff.
Okay.
Also, it has demonstrated power in like AlphaFold structural prediction. I think those people can apply to many different things.
So basically, you're saying it would help with all of these things, so it sort of supersedes everything. Okay, fair enough. Does everybody know what AlphaFold is? Just because you mentioned that, I want to make sure everyone's aware. Yeah, hopefully. Yeah. Anybody not know what AlphaFold is? That's maybe a better question. So when did that publish? That was this summer or last year? 20, so 2 years ago. Oh my God, that was a long time ago. So AlphaFold is basically Google's kind of, I don't know what moonshot project where they basically took the entire protein data bank, so all protein structures that are known, and they use machine learning to be able to predict from scratch a protein structure, which has kind of been a problem in the space for decades, right? Actually, more than decades, probably. So that's kind of a huge win for AI to be able to do that. And some people say that that's unlocked the ability to engineer proteins. One of the critical things that you need to keep in mind about AlphaFold is that at the end of their AlphaFold prediction, they always do an energy minimization function to get a better fold. So there are some thermodynamics and some other more traditional modeling needed to fit into that AI, but that's what that's the AlphaFold thing. So it's kind of a big, in the space of protein engineering, that's a pretty big event in the last couple of years.
Probably a certain Nobel Prize.
Exactly, yes. It's Nobel Prize. As CRISPR is Nobel Prize, AlphaFold is Nobel Prize, right? Okay. And it's free. So you can go on if you know how to code and use it. This will be one of my diatribes that coding is an important skill that everybody should have. Okay, so advances in synthetic biology. Who said that? Fair enough.
David Baker wrote Rosetta Fold, which basically forced AlphaFold to publish.
Okay, got it.
He submitted Rosetta Fold, and the AlphaFold guys didn't want to get scooped, so they published AlphaFold.
Got it. Okay. Good to know. I did not know that, actually. Systems engineering. Who said that one? Doug, why?
Second, biologic cells are complex systems, and as we move forward, we need to start thinking of that as complex factories that you can modify, change, optimize.
Okay. Anybody who said systems engineering that has a different rationale? I guess you maybe.
I would just extend that. I know a lot of the conversation here is on the engineering biology, but I think that goes beyond just biology. Right? At the end of the day, you have to put this in a manufacturing environment, so you need downstream process, right? You need an economic model. You need all these different things, all these different disciplines that all have to fit together. And even within the context of this, right, many of you could be perceived as tools, but all those tools need to work together to build a house, right? Or a manufacturing plant.
or a house. It could be the sustainable bioeconomy where the house is made out of some sort of bio stuff. Okay. Anybody want to add anything before we move on from this? Because I think this is very common. This highlights that there's a diverse set of opinions in terms of what is driving the field, and how do you define the field, first of all, and then what's driving the field, right? So how did I think about answering this question? So a couple of weeks ago, actually, I was with the chair of our department, and we were interviewing a really well-known cell biologist who was applying for a position in the Duke Biomedical Engineering department. And actually, she was looking for a partial appointment in the biomedical engineering department. And so the question came up, she's a cell biologist. She's got some cool genetic tools to change things about cell biology, and she's done a lot of work historically to be kind of one of the leaders in the field in terms of cell biology, in terms of different types of cell systems. But the question came up, what is engineering, right? Which I think sort of ties with what is the field definition, right? And it was an interesting conversation because I think there are a lot of folks who think molecular biology and genetic engineering is engineering, right? So you change a gene, you put it in the cell, and it's engineering, right? And I think the people who come from a hardcore engineering background, maybe like chemical engineering, would say, well, that's just genetic manipulation. That's not engineering, right? So from a departmental standpoint, our department struggles with this a lot. We've gone from, when I started at Duke, it was 18 faculty, something like that. Now we're like 32. And most of those additions are in cellular and molecular systems. And a lot of the folks who are hardcore ultrasound and imaging and biomedical devices are kind of like, those guys don't do engineering, they just do biochemistry, right? So this becomes a pretty routine question that's asked a lot, right? So the answer that the chair gave was that if you're an engineer, you are engineering a system for an application, right? Which is where I think that I came up with two answers to the question. So one that no one answered was new technologies. And the other one was standardized systems engineering, right? So I agree with Doug and Matt and in between there in terms of what I would say really is going to drive the field forward. And I'll kind of walk through, hopefully to convince the rest of you why I think that and why it's a good idea that we should focus on that as a group in the next several minutes, or if it goes longer, it goes longer. So new technologies. And what I mean by that is there's technologies developed all the time, and we develop a lot of new technology in my lab. And you really want to develop technologies that push boundaries. And this is important for lots of different reasons, but what do I mean by that? So if you think about some sort of result of performance, and this could be a cell output, it could be a fermentation output, it could be, you name it, it could be some sort of, anything you want. And you think about the frequency of getting that solution. So this is the sort of, there's a search space, here's all the solutions you can have in that search space, right? And you're searching the search space, right? So science is really about understanding that distribution. You learn about how it works, you observe it, kind of understand some basics. And then most technology development tries to optimize within that natural realm. Like you're going to push something to a certain optimum. Our field has historically been sort of swamped with technologies that do that. Take genome models, for example, and then push to optimize that genome model in the boundary conditions of that cell to make something, right? The problem is, this is how I define innovation, right? So how do you go from what is natural and engineering the natural system to jump outside of those boundary conditions, right? And that's really, really hard. And people talk about this a lot. This is one of my issues with machine learning. So if you need a solution that's out here and you're using machine learning on a data set, machine learning predicts within the data set. It doesn't predict outside of a data set, right? So you're never going to be able to predict the innovation. And that's how machine learning in general has been used in our field. Zymergen is a great example, I think, of this that didn't go so well, right? And there's other reasons why I think it didn't go so well. But, you know, and they've even presented a slide similar to this at a meeting, and then they stopped presenting that when they got negative feedback. Because they basically said, we have 1 million strain designs to make a product, and they all end up in that gray curve. And they have a couple that maybe go a little bit out, but they don't have anything that hits their commercial metrics because they're basically building what is sort of constrained in the system, right? So how do you get around this? So the one thing that's really great about machine learning or other science and technology development and other engineering strategies is that you learn about the boundaries, right? So if you don't know what the innovation is or how to design the innovation, at least you know what's keeping you in that box, right? Does that make sense, everybody? So the first thing is you can't get out of the box if you don't sort of clearly define what's in your way, right? What are the boundaries, right? So when I think about new technology development, you know, and there's some examples of systems integration, I think with Rommel presenting his poster over there and Adim more on this boundary pushing concept for DNA synthesis technology. It's sort of, if you define what's keeping you in, then you at least know what you have to do to kind of get out of that, right? And of course, there are some boundaries that you can't break, so you can't break thermodynamic laws or things like that, but there are some that you can. And then with systems engineering, right, I think that's also another critical thing that we need to think about. And I kind of put it both into what Doug said and what Matt said in terms of the system, how you define the system. So what is the scope of the system, right? So this is what NASA says, right? The system engineering is defined as a methodological, multidisciplinary approach to the design, realization, technical management, operations, and retirement of a system, right? So the whole life cycle of a system. And the combination of elements that function together to produce the capability required to meet a need, right? So Jasko's not here today, right? So yesterday in the car, for those who don't know Jasko, he's one of the board members of DMC. We were talking about, I've been teaching design courses for the last, too long, 5-6 years now. And, you know, what I think that has really hammered home for me teaching that is that when you're designing rather than learning, you're doing this bottom thing, right? Whatever you're making and building with engineering has to meet some goal. And if it doesn't meet the goal, you have to kind of reassess, you know, is the goal appropriate? Is it within the boundary conditions? What are you doing to hit that goal, right? And so I think that there is more than enough capability in the world today in our field to do this process, right? So going from DNA, mRNA to enzymes to single cells to cell culture to fermentation, it is possible, right? I don't think we need new technology to do this. Now, maybe we can do it faster, cheaper, better. I totally get that. And there are examples of this, right? So there are real world examples of doing this process successfully and even commercializing it, right? So it's like one of those memes that I was looking at memes to add to this talk, and there's a meme that's like, who's seen Lord of the Rings? Everybody? Yeah. So there's a meme that's sort of like, the wizard knows how to recruit eagles to fly them, but they're walking like hundreds of miles. What is the point of that, right? So we've obviously shown that we have the technology, so why are we walking, right? And I think the issue there is that historically in our field, the arrows have been one way, and that's how we think about things. That's how we manage things historically. Still to this day, you hire people into a system and you're expecting this kind of workflow. You start at the bottom, DNA, RNA, pathway, cells, and then when the cells are good enough by some definition, you optimize the process around them, right? That's kind of the workflow, how it's been for the last 30 years, right? And so if you're actually thinking a systems engineering viewpoint of biomanufacturing, you have to think about going top down and how do these systems interact with each other, right? So a systems view, about another view of that is sort of this cog view, right? Where you have to consider when you're building the strain, you have to consider how it interacts with the fermentation, how it's going to scale up. You can't just build a strain and then try to scale it up. That people have done that successfully, but it becomes a very one-off solution. How do genes affect enzyme networks, strains, fermentation, scale up, TEA, right? So metabolic engineering, the goal of metabolic engineering is to do this, right? And so where has that really been over the last period of time? So it's a pretty young field. I mean, I know many of you were not alive for most of this, but from a scientific field perspective, this is pretty young, right? So I think it was 1991, 92, maybe there was a Bailey Stephanopoulos paper that kind of coined the term metabolic engineering. There were probably in the 80s, there were some studies that did more kind of metabolic engineering proof of concept work. But that's kind of when this field kicked off as a field proper, right? 1990. That's not that long ago. It's only 30 years, right? And during that time, and these are not necessarily in order of appearance, but in terms of order of maybe conceptualization, there's been a lot of fundamental work in metabolic engineering. And I think one of the issues in our field as well is that we don't teach this. Right? We have a lot of metabolic engineers who are doing metabolic engineering in practice that have never done metabolic flux analysis, have never done metabolic control analysis, maybe don't even know what those are. So I'll ask the question here, who knows what, besides Doug, who knows what metabolic flux analysis is, and who knows what metabolic control analysis is, and who knows what the difference between the two is? Anybody? Matt had a hand up and then he put it down. Anybody else? Okay, so, these are mathematical constructs that are basically built for pathways that were kind of developed from a theoretical perspective early on in metabolic engineering. And one of the issues with them is that they weren't really predictive in many cases of what happened at the macro level. So people kind of went out of favor, although there has been some work, ongoing work, obviously, with C13 flux analysis and isotopic flux analysis all the way till today, and even control analysis is still used. Although I think people rely too much on synthetic biology when they're actually doing metabolic control analysis, they're not actually doing the math anymore. And then there's inverse metabolic engineering, which is kind of what I kind of started doing when I got into this field, flux balance analysis, which is different from metabolic flux analysis. I know it's confusing, and genome scale models. So where we went from metabolic flux analysis and metabolic control analysis, those are ways of sort of mathematically modeling fluxes through networks, pathways, and then flux balance analysis came on, and sort of Paulson sort of spearheaded that whole thing. And that was, how do we model the entire metabolic network from a gene structure? So we have the genes, we can go genes to enzymes, and we can model the whole metabolic network, right? And so if we can model the whole metabolic network, we can kind of understand and design how to put it into whatever sort of state we want to do. And along that came with how do you build those models, those metabolic models of the whole cells, right? So the genome scale models. So now you can actually go to any database, download the genome, and I think it's the GEM server. I don't remember what it's called anymore. Maybe they upgraded it. You go to that server, and it builds you a metabolic model of that strain, and then you can do flux balance analysis on any organism you want. Right? I mean, we can do that. Like, there's no development needed. You can go on. I mean, you have to download stuff and upload stuff and know what you're doing. But I mean, that's the state of the field. And that's the state of the field a while ago. Then we had some of the issues that were brought up with genome editing, right? So until maybe the 2000s, we were still doing gene editing even in E. coli the old way, right? Then there was something called recombination came up, which kind of opened up a huge bolus of how we can do gene editing and how we can make modifications in organisms. At the same time, there was the omics era, which was sort of going to solve all our problems, because we could learn everything about the proteome and the transcriptome and whatever ohm, and that was going to tell us everything, and it was going to tell us how to engineer stuff. At that same time, we sort of developed things like OpNoc. Who knows what OpNoc is? Again, maybe only two people. Yeah, I don't know what it stands for either. But basically, you can go into that genome scale model, that metabolic model mathematically, and there's a sort of a linear programming algorithm that was developed that allows you to figure out which genes to knock out to optimize production of your product, right? So we can do that. That's formulaic, right? You can go in, you can download these things, you can do OpNoc, you can get your system designed from a metabolic network, right? That's kind of Genomatica. So Genomatica started off with that technology platform. They've obviously gone past that since then. And around that time also, we had the first sort of rational protein design capabilities for different enzymes. In the last decade, we've seen a lot of machine learning, automation of molecular biology, automation of laboratory evolution, so ALE type programs, and then of course CRISPR editing came recently, and of course dynamic control came in 2013-14 timeframe, right? And I say dynamic control as a broad class, and that's important for the rest of the group to understand. I mean, DMC is the name of the company, but dynamic control is a technology platform that's much more broad, used in different ways. The term is used in the space in different ways than when we talk about it, right? So people can dynamically control pathway fluxes and regulation, and our dynamic control is really, I call it in the group, two-stage dynamic control, because we're actually switching. We're not just switching stuff inside the cell, right? So there's all this background, and all these capabilities that we have, and yet there are very few, we've done so much work, but there's very few examples of really success stories of going from, there are some, but there are rare ones, going from concept to a product, right? Oh, and AlphaFold also. Sorry, I forgot to put that one in. That's another recent advance that's really amazing. Okay, so an OPX moment. So this is me and the CEO of OPX back in, I don't know when this was taken. So we developed an omics technology and sort of an inverse metabolic engineering technology to kind of understand how strains will evolve when they're making a product. And that was some of the technology that we founded the company OPX Bio. And I remember having a conversation with him about when he joined the company as a CEO about what we needed to build for the research team. And I said, well, we need to build some, molecular biologists and some fermentation folks and some strain stuff. And he kind of looked at me and said, well, what does this omics technology do if it doesn't do all that, right? So I think in the space, one of the problems and why I think systems integration is really important is there's a thought that there's a magic bullet that's going to solve everything. Like one of these things alone is going to be the thing that gets us to the finish line, and that if we have any single technology, that means we don't need any other technologies, right? Or we only rely on one technology. And it was a pretty uphill battle with Chaz Eggert to be able to sort of expand the needed resources in the R&D team, because we were bringing an advanced technology, but that advanced technology by itself was never expected to be the only technology that we used. But there was an assumption that it was going to be the only technology that we used. So I think we have to be careful of that when we think about advances in technology. So how does the two-stage dynamic metabolic control platform fit into this historical context? So if you haven't seen this, hopefully you have. This is how it works, right? So the general schematic. So you have cells grow up, they consume some sort of nutrient, at least in the first implementation of the technology. And when they are done consuming that, they go into stationary phase, and then you can dynamically turn on or off metabolism and make your product of interest, right? So the green line is making your product goes up in concentration, and the red line would be you're turning off certain things that you don't want in the cells so that you can make a lot of product. So one of the things that we did when we designed this technology is we were thinking a lot about this problem, right? So we had a set of boundary conditions, that if you think about it, all of these technologies are searching in that gray space, right? They're optimizing the genome in the gray space. They're developing pathways in that gray space. They're looking at metabolic control in the gray space. So there's the natural space of how biology works, which is this space. And almost all metabolic engineering that had come before DMC, and we'll see if DMC holds out as a two-stage DMC to really prove this point, we're really focused on optimizing in that box, right? And so one of the things when we were thinking about DMC development and what we could do is we were thinking about what is wrong, what are the boundaries, right, that are causing these limitations in biosynthesis. And so if you think about it, and you do all these, and you learn about all these genome models and all these metabolomic studies, for example, you would say that intermediate or metabolite A in any cell line has a boundary condition between 10 micromolar and 1 micromolar, or 10 micromolar and 0 micromolar, or 100 micromolar. I guess it's up and down depending on your view, right? So there's a small range of conditions for any of these intracellular metabolites that sort of exist within cells, right? And so, but if you think about it, you do an enzyme assay or you do something in vitro, you can change those concentrations arbitrarily all you want to hit some thermodynamic limit. So the question was, you know, we're artificially constrained in this gray space because biology has evolved for itself to be adaptable and to grow, right? So there are conditions and constraints on sort of the network and the metabolism, right? But how do we then get out of those constraints was sort of the idea, right? So dynamic control, turning off central metabolism, turning off metabolism was one way that we were going to experiment with to say, if we can completely have cells that are no longer growing and shut off a bunch of stuff and deregulate stuff, we should be able to push these levels of things in the cell way beyond what their boundary conditions are in normal conditions, right? So we know what the boundary conditions are. We know sort of how they're regulated, right? And what is controlling that gray space. So if you're stuck in the box, the first step is mapping out what you want, but rather, how do you get out of the box? So we didn't necessarily anticipate the DMC technology was going to generate the improvements in performance that we've seen, but we were going for how do we get out of this regulatory boundary condition regime that is defined by the way cells act and the way they grow. So one of the important things about this is it's not, you know, everyone, I get a lot of comments about the synthetic biology involved in DMC technology, which is the switching technology and the CRISPR technology. But one of the fundamental benefits of this has nothing to do with how you do the switching. It's with the fact that you can actually get outside of the box, that you can actually engineer a metabolic state that's not boundary condition of growth, right? So stationary phase or non-growing cells, and then able to sort of manipulate central metabolic enzymes is really critical to that. And there are probably a million different ways you can do that, right? You don't have to do it exactly the way we do it, right? But synthetic biology is cool. So if you publish something, you have to play up the synthetic biology and the switching and the circuits, right? So Jisha was part of, we had a postdoc, John Berg, who led a paper, this is figures from, and Jisha actually contributed quite significantly to this paper. So if you look at getting outside of that box, right, and you look at, theoretically, if you could produce a product at a specific rate, which is the up axis, right, so this is the average specific productivity, right, and you think, and you just take the boundary conditions off these models, and you say, these concentrations can go whatever you want them to go to be able to force really high fluxes, right? Sort of like thinking of an in vitro technology. You can predict that they would go really, really high, but if you look at all the dots on this plot, right, these are all real world results, which are bounded by the conditions of cell metabolism and growth, right? So on the one axis you have yield, the other axis you have the sugar uptake rate. So this is, you know, obviously you can only make as much product as you consume sugar, right? And then the top, the up and down axis is how much product you make, right? So there's a theoretical constraint if you take the boundary conditions of biology off, that's a lot higher than when you put the boundary conditions of biology on. So one of the things that we were trying to go for, I mean, this is a review article, but one of the things we were trying to go for is that if we do two stages of dynamic metabolic control correctly, how can we access that top number, right? How can we push past the boundaries that existed before? And in some cases we have, and we've learned some new biology along the way. So this is actually a paper from, I guess, a couple years ago from our group, making xylitol from xylose. And I won't go through all the metabolism because I know a lot of people here aren't really metabolic people. But what I would say is this entire piece with this SOXRS and this molecule here is negatively regulating this molecule, which is then positively regulating this enzyme. That was not really known to be active in E. coli, right? So we did some switching. We pushed things beyond the boundary conditions of what normally exist. And when you go outside of the boundary conditions, new metabolism kind of arose as what was going on, right? Which was extremely interesting, right? And because of that new metabolism and deregulation, we were able to get much higher fluxes than we anticipated of xylitol from xylose. So this is an example, and actually in a lot of the work that we've done, we find that the things that give us the best results are things that dynamically deregulate metabolism, so then you can have really high levels of metabolites that then drive really high levels of synthesis, right? And so beyond what would be possible if the cells were growing. So that's sort of how the DMC technology, or the two-stage DMC technology, sort of was envisioned and how it's been operating to kind of push outside of the boundary conditions that have existed in the field for a long time. But the other thing that we did when we designed this technology, because we did design this technology, is we wanted to build a system that was integrated and intricately tied to other parts of the process, right? So instead of going from enzyme to strain and then building a bioprocess around the strain, we, as you probably have gathered, maybe it's not as, the beauty of it is maybe not as well observed, is that this is completely tied together. So the genes, the enzyme network, the scale up, the fermentation, and the strain are all designed together. The process was designed to work with these strains. The fermentation process was designed to be robust to scale up, right? So it's a system that is of limited scope. It's not going to work under other fermentation conditions, for example. But it's a two-stage system with a standard fermentation process that the strains are co-designed for, right? So this is a part of the systems approach or systems integration that I think is critical in the field, because if you're going step by step by step, then every time you do another step, you have a new set of discovery and a new set of things to do. And I think that historically that was kind of the way that we operated, and it worked, occasionally better than others. But I think we know enough now, and we have enough skills now, and we have enough tools now that we can start to build systems like this, right? So that's basically this process that we say. So it's a simple process, but everything was designed around this process. So we started with, what did we think would scale up? What did we think would be robust? How do we design strains to fit in that? How do we also design strains that can go past the boundary conditions? And then how do we go back to even the enzyme networks and genes? Like we have standard promoters we use for expression, things like that. So we've kind of gone through, and I know I've been going long on time. How do we define next generation biomanufacturing? We haven't answered that yet. A historical perspective of metabolic engineering, and then the two-stage dynamic control and context. I bring that up because I know, I'm going to pick on Doug again, but many times he'll say, you guys have an E. coli, you have an E. coli technology, and that is true. But I think the thing that is missing in our communication is this and this, right? So how do we communicate and sort of position ourselves to take advantage of the systems integration and sort of outside the box rather than thinking about it as a synthetic biology technology? So what's next for biomanufacturing 6.0? So Lynch Lab future directions. So obviously we are going to continue working on strain, fermentation, scale up, TEAs, which we've been building, genes, different products of interest to different partners. And we're also going to start working on models. We have enough data now that we can start to build models to predict which strains to use for different types of products, and how do we actually model genes to expression levels. And of course, we have some work. I think Mike has a poster on the right, on my right, on enzyme engineering and how do you think about search space for enzyme engineering. And then, of course, there's system engineering. How do you put all this together? We also, I'm spacing on. We submitted a DoD proposal for that biofoundry at one point, and we tried to brand this in a certain number. I forget what the name we used. SHIP. What did that stand for? Had two P's. I think it was scalable host integrated processes or something like that, to kind of say the fact that, you know, these strains are integrated with the process and they're scalable, et cetera. They're not separate. You can't really separate them from the process. And so we're replicating that with additional host strains. So these are some preliminary results with Saccharomyces, which is pretty difficult to work with. So what you see here is cell growth on the left, and then fluorescence protein expression on the right. So again, in this case, we're just trying to make sure we can turn things on and off. You have the case where you have high phosphate, where you don't turn something on, and then you have the case where you have low phosphate, which is consumed completely, and then you can turn on your gene of interest, right? So we have the switching working in Saccharomyces. And then we can actually, which is pretty cool, but not very impressive from a production standpoint, we've actually been able to get an aerobically growing Saccharomyces. Doesn't make any ethanol during growth, so it's completely dedicated to growth, which is what you see here. And if you're really interested in how, it's in one of our patents. And then only when you switch into phosphate depletion in the stationary phase, then you can start making ethanol. Right? So it looks very similar to the E. coli process. And then, of course, you can increase the yield to ethanol, which is the last bar on the right, when you phosphate deplete compared to when you did not phosphate deplete. Why I bring this up is this is not just a E. coli process, because it's not really a process, it's a system or an approach. But there are definitely challenges. So Saccharomyces, for example, it's really hard to get Saccharomyces not to ferment and grow aerobically. That was not an easy challenge. I mean, it looks easy now, but it was really hard. And it's actually really hard to get Saccharomyces to produce anything in stationary phase. They don't like to metabolize in stationary phase like E. coli. So there are challenges. If you move this system to other organisms, the concept, I think, will work and hold, but there's different challenges on the metabolism, on the biology, to get it to work in different systems. Okay. So DMC. So as I kind of added this discussion from yesterday, in response to a lot of what was discussed yesterday. So some of the things that have to get added, right? The technology part on the left, which is the genes, the enzymes, the strains, the fermentation scale up, and then the techno-economic analysis. But then there's two really important things, which is product picking and commercial strategy, right? And so they have to be integrated if you're going to systems integration with everything else, right? So I think historically in our field, and I see that we do this a lot, and I'm not saying that it's a bad thing or a good thing or blaming anybody, but I'm saying that this is the way it has worked. So this is the model that is easy to follow, is either we do the top thing, which is we say, let's define a commercial strategy, then we pick the product, and then we figure out what technology needs to do that, right? Which is sort of an approach we kind of took at OPX. So we said, a really good opportunity was industrial large-scale chemicals. So then we said, okay, the best product in that is acrylic acid. And then we kind of defined the technology needs from that, right? Which is one way to do it. The other is sort of the technical capabilities, which is we can make 4 things, or if you have a technology that only makes one thing, it's an easy decision, right? And then you product pick from that, and then you develop the commercial strategy. And I think we're kind of in a mixture at DMC of both of these approaches, right? So the question is, how do you actually integrate this? And take advantage of the technology for your product picking and commercial strategy. And I don't necessarily have an answer, but I think that developing that process to better integrate things will lead to better results. So if we can do that, you know, I think we meaning Matt and I back a couple of years ago, were like, well, how do we want to brand DMC? And it was like, the sustainable BASF or something like that. So think about a large chemical company that sort of dominates the space in terms of producing materials that the world uses. If that's the vision that the company really wants to grow to that level, to be able to make a lot of stuff and really change the world, we got to get this right, right? And the technology, I think, is there already, assuming that we actually have the product picking and the commercial strategy, right? So that's my 2 cents from yesterday. So some final thoughts. So one final thought is biomanufacturing 6.0 is not something that's very sellable. So I will turn to the group to think about, in light of this conversation, if you're going to brand something that's exciting, to say we're going to now be able to do this because we can do systems integration, or we've developed some new technology, we can do systems integration, what would that be? Biomanufacturing 6.0 is definitely not a sexy term, as well as systems engineering is also not a sexy term, right? So 2 things that are not. But if you think about other engineering disciplines, right, you only get to products when you start doing systems engineering, right? You don't have computers if you're only working on a circuit. You don't have, you know, space shuttles from the NASA example unless you have all the parts together. So historically, our field thinks that, or not thinks, but there's been a perception that we're going to have one technology that's going to rule them all, and then we can develop that and commercialize. But the reality is, if you look across all other engineering and science disciplines, unless you do systems engineering and systems integration, you never actually end up with a product. So it's not sexy, but I think we need to figure out how to change that so that we can push the field forward in that direction. So I'll pause there and see if anybody has any thoughts on terms that are exciting rather than my really weak terms. And I know I've gone way over time, so I apologize. Nobody, no thoughts.
Food for thought for the remainder.
Fair enough. Any questions?
I want to make a comment. So I know you recorded this, and first of all, I thought it was a brilliant talk. I loved it, and I think it's the sort of thing that, you know, would be great if DMC could publicly post. Because I don't think you said anything that was confidential, and maybe some things.
Someone should check that. Yeah.
I didn't want to say anything until we were at a
Fair enough. Yeah.
where we could actually cut out my comments from this, because I certainly don't think they should make it there. But I think, because I think, you know, this is kind of like visionary talk that we need to have out there as we take it to the next level of growth. So I just want to throw that out there, because, you know, this is one of the reasons why I was interested in joining the DMC board as an independent board member, was because I believe in this, and I told several of you that, you know, even as a grad student back in the 80s, we were sort of dreaming about this, but didn't know how to do it. So when I saw Mike and Matt and team do it, it got me excited. The second comment I want to make is, you know, you're talking about kind of going outside of the box. There is another technology that is also kind of going outside the box, and it's you know, struggling away, but it's a cell-free system.
Yep.
Jim Swartz is doing.
Yep.
And then Jim Swartz at Stanford is, I think, one of the thought leaders who basically is famous enough that he can do what he wants to do and doesn't have to worry about, you know, operating in that Gaussian curve, because you can also, you know, radically start changing things by doing cell-free, but it's got other constraints to it.
Yeah, no, I think that, you know, I think in the same article that had the sort of, what do you call that, the mountain plot, right? It talks about cell-free conceptually is amazing, but it's really expensive because you have to add everything, right? So the cell's already adding everything. So we thought of DMC as sort of a bridge between the cell-free and traditional fermentation for that exact reason, right? Because I think that's the cool thing about what they've done in that technology, is they can really push things pretty far. It's just expensive.
Just to let everybody know that Jim Swartz has a ARPA-E grant and is actually an advisor to an ARPA-E grant. It's not commercial, but so I am trying to learn as much as I can about cell-free, and there's definitely you know, overlaps between the two approaches.
If anyone hasn't seen it yet, Mike has a really good paper that positions the landscape of technologies from traditional wholesale bioprocessing on one end to cell-free approaches on the other. It's a really good paper that summarizes.
I think, you know, as the DMC is looking to kind of elevate our game in the social world, you know, posting a couple of those papers of Mike and a talk like this, I think it would do wonders in terms of, you know, both fundraising and awareness and even recruiting.
Yes, so you have a question in the back. Go for it.
So I'm not by this, so it's probably very nice. But when you talk about your aware of innovation, I actually was in the camp of the system engineering, so I'm happy to and because when you look at some of the graphs, everything that happens is very complex and everything influences everything else. And so it's got me thinking of, you know, how to talk about in collaboration with like systems engineering professor, and I think about John Sturman at MIT, who's one of the most brilliant minds in systems engineering. And if you think about what about a collaboration where actually he has all the optimization algorithms for systems engineering, and what we do in biology.
Yes.
But I just putting out.
No, it's a good idea. We haven't. So I think that would be a great idea. So the secret problem that you have as a faculty member is, even systems engineering isn't sexy to like the external public. It's also not sexy to recruit students to say you're going to do systems engineering, right? So those are good collaborations, but it's also how do you support them? Yeah. Oh yeah. No, that's true. Yeah. Yeah, fair enough, fair enough. So he can do all the work. That's great. Yeah, well, that'd be great. Yeah, that'd be great.
And I love that comment, because I've often thought, if we can figure out who are like the world's leaders in designing the factories of the future, you know, like the automotive factories of the future, and learn the techniques they use and combine it with people like you who understand cells, there's like a totally unexplored space there.
Yeah. No, I agree. Yeah.
Ways of thinking and combine them together. I have a video that I will send you if I can find it.
Perfect.
It sort of stimulated my thinking in this area. So I'm going to make a note.
No, but you're right. There's a whole discipline, right, of systems engineering, right, that has not really. They're never. Yeah. There's no. Yeah. No talking. Yeah.
Probably one of those people.
Carolyn, I think you had your hand raised a little bit back.
My question to you is, so, given, today, is recognizing the need that push the boundaries of vision, create more innovation, to be able to do it. Because if you think about what we would like to do with trace of biology, is ideally like create this range. So how far can we go today, and what more do we need to do to be able to produce a broader range of products?
Yeah, so I think that putting my systems integration hat, although I'm not a faculty or professor in systems integration, system integrations or systems engineering. So I think the challenge is the gears have to spin at different, there has to be a differential there, right? So I think that what is the commercial strategy with what we can do today, right? So that we're not picking things that require that optimization, as you said, because there are a lot of potential things that could happen, but those gears have to turn at a different rate than the commercialization, right? So I think if we do a one-line thing and say, this is a project and we have to optimize this and push the boundary to get that to work, that's probably not the best way to go about it. I think the best way to go about it is to ask those questions about what kind of products can we not do today, and what kind of boundaries do we need to go past to be able to do that. Have those gears turn independently, right? To say, here's how we're going to push past those boundaries. And when they're ready, then you go after those products, right? So I think there's a lot of possibility, but I think also there's a lot of products that we could go after right now, right? One of the things that I think that, I've talked to Matt about this is, this system is really well set up for bioconversions, for example, where you have limited pathway steps that take a different feedstock and you're upgrading it to a higher value product, right? That's actually not as hard at all as the metabolic engineering. It's pretty easy stuff. And we particularly have we have strains that have like high cofactors and high, things that can actually do those steps. So there are a lot of technologies like that, products that could fit into that technology that would be technically really easy and you could turn really fast. We have the things that are, that we've picked to date, the larger volume commodity things. And I think the challenge there is, even if the technology works, it's a couple of years, right? Building plants, finding partners, that commercial strategy, right? Maybe needs to be, it has to be recognized that that's more out of our control, and it's not something that, we can turn as fast. So putting all the front stuff to address those types of products is kind of a waste of the front stuff. Does that make sense? So I think there's a lot possible. I think it's just, how do you structure the work so that you're not, that you're focused on what the outcomes are for each of the individual things, and that they're talking to each other at the right differential. Which is not that I have a solution of how to build that structure. That's just what I think needs to happen. Because that's hard. Correct. Also the technology curve, which goes the other way. So where do they cross is kind of how I visualize that, right?
And then also think about, defined commercialization. And focus on those things that we can commercialize faster rather than longer, with less capital rather than.
Correct. Yeah. I mean, that's the textbook way people say you should commercialize new technology. And I think that the reality is, even, when we, to be really fair, right, when we designed this, right, we kind of had in mind TEA, scale up, fermentation, strain, but we didn't integrate it into, well, it has to be good enough to go through a tolling step before it goes to a commercial step, right? I mean, so that part wasn't really in the design criteria to begin with. Looking backward, so if everybody doesn't know this conversation, so if you want to commercialize a product, for example, that's at large scale, and you have metrics that are good enough to make money in a plant that you build, one of the challenges is how do you build the market and sell product before you actually get to that plant, right? So if you go to a plant that's already existing, the problem with that is that they charge a lot. So then your numbers that work in your plant don't necessarily work in the tolling model. So there's a gap between going from your proof of technology to commercialization because we didn't plan it, to be fair. That's why it's there. Because you could easily put it in the loop and say, we actually need to have 2x the performance, right, to make it to the tolling gap or what. I mean, you can design those things ahead of time. They just weren't incorporated in the original sort of way we were trying to integrate things. But I think we can learn that now and kind of figure out, what are those situations where we can actually get good enough to get over the tolling gap, right? Go down the cost curve and up the technology curve, right? Yeah.
And then as you build, you can actually put a big plan, you've got the economic.
Yeah. Yeah. But even planning that takes integration from the beginning, right? Which I think, to be very blunt, we didn't do. So. And I think we're realizing that now. Yeah, which is totally fine. Yep. Sure.
Thank you, Mike.
Yep.